Assalamualaikum warahmatullahi wabarakatuh everyone. Welcome back to my class again. And before we start, I'm gonna ask you, how's your holiday? Is it good? I hope you're all doing okay during holiday, because today we're gonna start our class. So before we start, I'm gonna introduce myself as usual. My name is Erfan Danish Erwan, and you can call me Sir Erfan. But before we start our lesson today, I want you all to watch this video first and think carefully about that video, because I'm gonna ask it later. So, what is your perspective on that video that have been shown? Take a moment to think about the different elements that you can observe. Now, I would like to highlight the 3 key points that I can saw from the video. The first key point that I can saw is the gated opening of the zoo. We saw the main entrance of the zoo allows people to enter or exit the zoo. This is much like a control gateway, similar to how certain structure regulate what enter and exit a specific area in a cell. The second key point that I can saw is there are many cages in the zoo. The zoo had numerous cages to keep animals in a specific area. These cages act as a boundary that protect the animal and also keep them within a certain spaces, much like a compartment in a cell. And last key point that I can saw is the fluid movement of people. Throughout the zoo, people will move freely, flowing pathway that connect to different areas. This movement was directed and orderly, much like traffic in a city. These 3 observations that I can saw can provide us a useful analogy to understand our topics today, which is the structure of plasma membrane in the cell. Just as the zoo had control entry, compartment and also directed movement, the plasma membrane also provide us with similar features that we will delve deeper on today's lesson. For this day, we will enter chapter 3, movement of substances across the plasma membrane, and we will learn about 3.1, the structure of plasma membrane. At the end of this lesson, you all should be able to justify the necessity movement of substances across a plasma membrane. Describe the components of plasma membrane and also its function based on a fluid mosaic model. And lastly, draw and label the components of plasma membrane based on the fluid mosaic model. Now, we will going to briefly discuss the key characteristic of plasma membrane. The first key characteristic is selective permeability. The plasma membrane is selective permeable on what can go in and what can go out, just like the zoo video, meaning it only allows certain substances to pass through. This selective nature will ensure that only essential molecules like nutrients will enter the cell, while waste products are expelled. The second point is size, polarity and charges. The movement of molecules across plasma membrane is determined by their size, polarity and also their ionic charges. Small non-polar molecules like oxygen, carbon dioxide will easily pass through, but larger and polar molecules such as glucose or ion will require specific transport protein to cross the membrane. The third characteristic of plasma membrane is its dynamic and fluid structure. The plasma membrane isn't rigid or static in a place. Instead, its components are constantly moving, creating a dynamic and fluid structure. The fluidity is crucial for the membrane flexibility and also functionality. The fourth characteristic of plasma membrane is movement of phospholipids and proteins. Within the membranes, phospholipids and protein are free to move sideways or laterally. This movement will contribute to the membrane fluid characteristic, allowing it to adapt and respond to changes in the environment. Do you remember the fluid movement of people in the zoo in the first video that I showed you earlier? If yes, then good for you. And the last characteristic of plasma membrane is the mosaic pattern of protein. The plasma membrane is often described as fluid mosaic models because of the various protein embedded within it. We have a channel protein, carrier protein that we will learn later. This protein form a mosaic pattern with each specific function such as transporting, signaling and also for structural support. After this, I will show you a video about an egg experiment. Please focus because it will help you to understand more about the characteristic of plasma membrane. So, guys, what actually happens in the video? Here's what actually happens. When the vinegar is added, the shell of the eggs is dissolved. The vinegar will react with the eggshell, causing it to dissolve and leave behind a soft and rubbery membrane of the eggshell. Secondly, the egg will swell. The eggs will absorb some of the vinegar through its membrane and it will swell slightly. Now, let us think about this experiment in the context of cells. Flexible barrier. Just like the eggshell dissolve and reveal the flexible inner membrane, our cells have plasma membrane that is flexible and surround the cells. Just remember the membrane of the egg just like a membrane of a cell. Secondly, selective permeability. The egg membranes allow vinegar to pass through but keeps the eggs intact, similar to how plasma membrane control what goes in and what goes out. By looking at the egg experiment, we can get a glimpse into the fascinating world of plasma membrane, which plays crucial roles in protecting our cell and also regulate our interaction with the environment. Now that we have this analogy in mind, let's dive deeper into the structure and function of plasma membrane. So, let's go deeper into the structure of plasma membrane. Plasma membrane consists of four main structures: phospholipid molecules, protein molecules, carbohydrate chains, and also lastly, cholesterol. So now guys, let's jump into the first structure of plasma membrane, which is a phospholipid molecule. A phospholipid molecule is made up of two parts, a polar head and also a non-polar tail. The polar head is hydrophilic, meaning it is attracted to water or water-loving. This part will interact well with the aqueous environment, both inside and also outside of the cells. The second part is non-polar tail. Each phospholipid molecule has two polar non-polar tails that are hydrophobic, meaning they are repelled by water or water-hating. These tails prefer to be away from water and interact with each other inside the membrane. In the plasma membrane, phospholipids are arranged themselves into a bilayer. Hydrophilic head face outward towards the water, while the hydrophilic tails face inward away from water. This arrangement forms flexible yet stable barrier that protect the cell and control the movement of substances in and out of the cell. Let's go into the second structure of plasma membrane, protein molecule. In plasma membrane, there are two main types of protein molecules. The first one is channel protein and the second one is carrier protein. Channel proteins are like gatekeeper with channels or pore that allow only specific ion to pass through the membrane. Think of them as a specialized tunnel, ensuring the selective movement of ions across the membrane. For the second protein, which is carrier protein acts as a carrier, transporting molecules such as glucose across the membrane. They can change shape to facilitate the movement of molecules from one side of the membrane to the other. Imagine them as a flexible vehicle, adapting their shape to accommodate and transport specific molecules. Both type proteins contribute to the plasma membrane functionality by regulating the passage of substances in and out of the cells. Channel protein provides selective only pathway that ion can move, while carrier protein facilitates the movement of molecule by changing the shape. Imagine this a protein, it can change shape to allow substance move in, and it can change shape to allow substance to move out. These are the differences between a channel protein versus a carrier protein. In terms of transportation, channel protein only transports small molecules and ion like oxygen, while carrier protein transport large molecules like glucose. In terms of the movement, channel protein transport it down concentration gradient, while carrier protein transport it down and up the concentration gradient. In terms of characteristic, channel protein has an opening in the center and it can be gated, while characteristic of carrier protein is it changes its shape to allow the molecule to pass through the membrane. Now, let us move on into the third type of structure in the plasma membrane, which is carbohydrate chain. We got two types of carbohydrate chain, which is glycoprotein and glycolipid. A glycoprotein is a protein which has a carbohydrate chain attached to it, while for a glycolipid, It is a lipid which has a carbohydrate chain attached to it. There are many functions of glycoproteins and glycolipids. The first function is they can bind with hormones and they can function as receptor to hormones such as insulin. When they bind into a hormone, they will trigger a chemical reaction in the cells. The second function is they help to stabilize the plasma membrane structure by forming a hydrogen bond with water molecules around them. And lastly, the function of glycoprotein and glycolipid is they function as antigen to help cells recognize one another, and also they function as a cell identification. Now, let us jump into the last structure of plasma membrane, which is cholesterol. So, cholesterol molecules are found in the plasma membrane. It is inserted in between the phospholipid molecules. So, the function of cholesterol is it can make the phospholipid molecules become more flexible, it can strengthen the phospholipid molecule, and also it make the plasma membrane become less permeable to ions and very small water soluble molecule because the cholesterol has a hydrophobic regions. So now I want you to think for a second. Is cholesterol a very bad substances? No, right? Because cholesterol also serve a function in our body. Okay class, we have reached the end of today's lesson. But before we dismiss our class, I want to recap the main point that we have learned today. Firstly, the characteristic of plasma membrane consists of selective permeable and semi-permeable. It only allows certain substances to go in and go out of the cells. Secondly, plasma membrane are not rigid and structured, but it is in a fluid form. Just remember the key point of fluid. And lastly, the various proteins built in the plasma membrane are built in a form of mosaic, or we can call it a fluid mosaic models. Now let's move on to the structure. The plasma membrane has 4 types of structure. The first one is phospholipid molecule that consists of a polar head and non-polar tail. The second structure is protein molecule, which consists of carrier protein and also channel protein. The third molecule is carbohydrate chain, which consists of glycoprotein and glycolipid. And lastly, the molecule is cholesterol that are inserted in between the phospholipid bilayer. Now, before we end our class, I just want to give one moral value, which is we should appreciate every small thing in our body. We are a complex being. As we will learn today, there are many types of structure, there are many types that serve many functions, and we should appreciate every small details in our body. Please do your homework on tutorial book, tutorial 3.1.1, 3.1.2 and 3.1.3, and please submit it onto my table before our next class.